Faculty Voices. Brian D. Farrell is Harvard Professor of Organismic and Evolutionary Biology and Curator of Entomology in the Museum of Comparative Zoology. He's also the Monique and Philip Lerner Professor of the Study of Latin America. Welcome to Faculty Voices, Brian.
Thank you, June. It's wonderful to be here with you.
Today we'll be talking about mosquitoes and other bugs,
Great.
which you specialize in. Your latest work is taking you to Brazil.
That's right. Yeah, along with a postdoctoral fellow, Paulino Ribeiro, to the Fundación Osvaldo Cruz in Rio de Janeiro.
What exactly are you looking at?
Thanks to generous support from the Lehman Foundation, we're looking at the genomic adaptations of these little mosquitoes to their particular habitats that they live in. Now, these are a little bit unusual in the sense that they live and place their eggs only in little containers of water that naturally occur throughout Latin America and the old world tropics as well. So they might put their eggs inside of a pitcher plant, or it might be inside of a bamboo stalk, or inside of a heliconia bract, or inside of bromeliad, which are the relatives of pineapples. All of those kinds of habitats have little containers of water in them. And those are called, and the things that live inside those little containers are called phytotelmata. That's a term that really refers to the animals that live together inside these little containers.
So, why should we care about this?
One of those little container dwelling mosquitoes is Aedes aegypti, which lays its eggs, which could, is a major vector of malaria and dengue and other diseases. And it lays its eggs in the natural containers of water, not so unnatural, in cities that are found in pots of water around houses. They're found in tires, especially old used tires. They often collect little bits of water, and those mosquitoes will lay their egg in any and all of those places. So that's the applied reason to work on this. But of course, like musicians or folks who study in the humanities or other disciplines, there doesn't have to be an application, right? Most basic research doesn't have an immediate application. It's for the sake of increasing human knowledge. So that's our overall mission, and it's a mission shared with a lot of other disciplines. But in this instance, there does happen to be an application, and that's to look at how it is that these insects are very specific to those particular different kinds of container habitats. Some are very acidic, some have lots of nutrients, some are very poor nutrients, and others may be very basic, that is on a pH scale. They differ, and the insects themselves show great fidelity. They're very loyal to the particular kinds of habitats they use. So we're interested in why that is. So that's one piece of the puzzle that we're able to study thanks to not just the Lehman Foundation, but also the Museum of Comparative Zoology, which has an ongoing postdoctoral program of biodiversity fellows. And I can tell you that Brazil is about the best contributor of qualified PhDs to that program. I mean, I have one from France now as well, and we have them from Colombia and Mexico and just about every other country. But Brazil especially has been very good in producing highly trained naturalists, people who work on the kinds of things that we're interested in the museum.
You mentioned that there have been advances in technology that allow you to look at these. Could you talk a little bit about those?
Sure thing. Yeah. Yeah. So these advances are genomics. So these are the new ability to sequence the entire genome of a species within a few days, and for a relatively small amount of money, a few $1,000s. This was the moonshot, of course, of genetic research back in the early 2000s with the Human Genome Project, which Craig Venter then came in. So the NIH took one approach. Craig Venter, this scientist entrepreneur, came in, invented a new technique and did it faster and cheaper. But at any rate, as a result, now 20 years later, we're all able to do this pretty readily. We do it here at Harvard in the Bauer Center, which is in the Northwest Labs here, up here in the North Yard, and we can do it very quickly and in a very cost-effective way. So what that allows us to do is to identify all of the genes in a genome. That is the 4 to 5 billion base pairs of information. That's the blueprint for making an animal. And we can study what the different genes are that control their abilities to live in different places. And so that's what we're interested in doing.
Is this new for you that there's a public health aspect to what you're doing?
Let's see. I suppose it is, and I've long been interested in mosquitoes because they are what I call tiny consumers. So I work on lots of different kinds of insects and I'm interested in other kinds of life forms that everything from viruses to fungi to parasitic worms that live at the expense of a larger host. So I call them all tiny consumers because, in fact, what I discovered early on in my career was that they evolve in very similar ways. And I could tell you a little bit about those too. But that is the common thread that unites all of the things that I've worked on and the folks in my lab too. But I've been interested in mosquitoes in particular and their public health effects since I was a beginning graduate student and heard this person speak from NIH who gave a talk at the University of Maryland where I was and really blew my mind with the kinds of interesting things that he told us about mosquitoes. So I've always wanted to work on them, but I have worked on bark beetles, which are not a public health, but they're a public forest menace. And I've worked on other insects that attack crops and things. As the grandson of farmers, I've always had an interest in agriculture and in where culture meets biology and vice versa. So that's, for me, that's about the most interesting area to do research in. And so it's what unites everything I've worked on.
Let's talk a little bit more about how culture unites with biology. What's that cross section?
We are biological beings and we are conversing now using language, which of course it's a mix of biology and culture in the sense that we're born with an innate ability to learn a language, but we do have to learn it. So we're not born to croak like frogs are. Frogs don't need to hear and they don't need to practice. They basically are born to make the sounds that they make. And most mammals are that way and most insects are too, but songbirds are not. Whales learn their songs too, and so do seals and a few other animals, mostly songbirds though. So we're one of a handful of species that are vocal learners, and we're social species as well. So that serves our social cohesion need very well. It allows us to communicate with the details that language allows us to convey, but with the emotion that music allows us to convey. So those things together, really. So I'm really a little bit interested in all of these kinds of things, and they're all connected in one way or another through the biology of culture, which you gracefully edited a special issue of Revista years ago now.
Yeah, it was a lot of fun working with you on that. Mosquitoes have a special sound?
They do. They have a very high pitched frequency that the males emit to attract females. We can't hear it, but you can hear some of the lowest frequencies when you hear those mosquitoes around your ears at night. And the funny thing is that those are not the ones that are biting you. Those are the males, right? So they're not doing anything, but it's the females that they're attracting that you have to watch out for. Anyway, yeah, that is the acoustics of the mating selection in mosquitoes and lots of other animals, especially insects, but not only, make high-pitched sounds that we can't really normally hear without special recording equipment, which we use.
That's interesting. You said in a speech, going back to mosquitoes again, possibility that we may now study stasis and evolution, as well as rapid change, promises a new dimension of integration in evolutionary theory. As I mentioned to you before, I don't have a really solid scientific background. So could you explain that to me and to our listeners?
Yeah, well, thank you for asking the question, June. Yes, I gave a talk in Japan this summer on this topic, and it is one that I've held for a long time, actually, since I again was a beginning graduate student. I first noticed then that related insects feed on related plants. So I'm interested mainly in herbivorous insects that eat plants, and they are tiny consumers, and so they usually eat one kind of plant. I happen to be I have a grant now from the dean to work on milkweed beetles in Mexico. And all milkweed beetles eat only milkweeds. In fact, they eat different species of the same kind of milkweed, which is the same kind that we have up here in our fields and even around Cambridge. Other insects feed on other kinds of plants. There's a group of insects that only feed on cucurbits, things like squashes and cucumbers and the like. Others that feed only on tomatoes and chilies and their relatives, and others that feed still on grasses. And so it turns out that most insects really only feed on a particular kind of plant. And that was the first thing that I noticed. And I quantified it back then to ask how many changes in host plant type per speciation event, that is, when you're looking at the evolution of groups of species, how rapidly do they change host use, the kinds of plants that they eat? And I found not very rapidly at all, generally. There's some exceptions, which are also interesting. It has more to do with the plants than the insects. But at any rate, so I noticed that. And then a little while later, I noticed also that it wasn't just insects, but in fact, just about all tiny consumers, so whether they're viruses or they're parasitic worms or fungi or whatever, are also similarly what we call conservative in the evolution of using different kinds of hosts. Related species feed on related organisms. So I wondered why that might be. And I've wondered about it ever since, really. But I did discover along the way another similarity between tiny consumers, and that is not just the kinds of plants that they feed on, but in the parts of plants. And I noticed that was common among other tiny consumers too. For example, milkweed beetles all feed on the roots of plants. The adults feed on the leaves, the larvae feed on roots. Other kinds of beetles feed on only on seeds, maybe of beans and their relatives. Others feed inside trunks of trees. Others might mine the leaves or feed outside on the leaves. All butterflies do the same thing. They all feed on the leaves of plants outside as caterpillars. But if you look at their relatives among the moths, some are feeding in seeds, some feed on roots, some feed on trunks, etc. And so there is variation among, in host use, in host part use, but it's even more conservative than which kinds of plants they feed on. So that was the kind of evolutionary rule that I stumbled on for tiny consumers, that the rate of evolutionary change in what kinds of hosts is a little bit faster. It's very slow, but it's a little bit faster than that of which parts of the plants they use or which parts of their hosts that they use. You know this, if the last time you had a cold, you could self-diagnose it because it was in your nose. But you never saw a test. You never went in for a rhinovirus test. You knew because they invariably infect your nose. But if you'd had the flu, you'd know that it was the flu because it attacks the other end of your body. Or if you had some other viral disease, maybe it's the mumps, maybe it's chickenpox or whatever it is, because they're all very specific in which parts of your body they attack, even though the immune system is the same in all parts, right? So it's a bit of a puzzle. It's something that virologists have called tissue tropism. So I've stolen that term and I'm using it for all tiny consumers now. But the mystery has been why, right? And so now with genomics, we're able to finally, I hope, at least test ideas about the why. Now, believe it or not, 100 years ago, a very famous geneticist and theoretician named Ronald A. Fisher, he was a successor to Darwin who put numbers and mathematics behind evolutionary theory. He puzzled over how is it that in one place, in one population, we can have different morphs. That is, you might have blue ones and red ones and yellow ones all coexisting at the same time in the same population and through time. How is it that their genes for being red or yellow or blue aren't scrambled every time that there's a reproductive event? Because we started to learn a little bit about genetics. And we know it's a great scrambling game where the offspring are mixes of the parents. So why is it that those aren't all mixed up? So we thought maybe there's this genetic mechanism that maybe it involves inversions where you take whole parts of the chromosomes and you turn them upside down and put them back in the genes. And then they can't exchange and scramble with every time that there's a sexual reproduction event. Now we could go further, but it's more technical. But let's just leave it at that. So he came up with this mechanism called inversions. And so I realized that maybe that's the kind of thing that stretched over evolutionary time would capture the genes responsible for using different kinds of hosts or different parts of hosts. Mm-hmm. And so when my PhD student, my most recent one to graduate, Sang-il Kim from South Korea, from Seoul, he was actually, incidentally, a brilliant beetle guy who was born to the cloth in a way. He'd been collecting beetles since he was 7 years old and in the national newspapers for his discoveries in Korea. Fantastic, really. He came here to study, came to Choate, and then he came to Harvard as an undergraduate. He's the only Harvard undergraduate I've ever taken on as a graduate student. And so we've been together for 13 years. And now he's a postdoc on this Lehman grant. But at any rate, he said he's a master at mapping these genomes and figuring out which chromosomes the genes go on. And when he told me that we can do chromosome level genomics, I realized, whoa, we can test Fisher's hypotheses about inversions and the like for the first time. And so at any rate, I'm really super excited about this. This was just a year and a half ago that this became possible. So that's what we're doing, is looking at rates, evolutionary rates in host use and other things too, using this chromosome level genomic analysis. And so now you've got the state of the art in the MCZ and in evolutionary biology.
That,
That's where it's going. And I'm very excited about it.
that's really interesting. Now, so how much of this work is taking you out into the field and how much is in the lab? And what does that field work look like, particularly with mosquitoes?
The field work is, fortunately, I don't have to do much of the field work because we have Paulino Ribeiro from Rio de Janeiro here for the next year. He's been the postdoc who's been studying these very mosquitoes for his PhD. And he's the expert. He just got back from Brazil. He collected 3,000 specimens in 10 days' time. So I could never hope to help. Even I could carry his vials for him or the net, but this guy is really the world expert on these mosquitoes. So he doesn't need my support in this, except with the conceptual part that we're working on together. So in fact, he's been down to Colombia. He's been here collecting with our local mosquito expert down in North Carolina and here in Massachusetts. Because these little container nesting ones, they're here in mass. There's one species in North America, and it lays its eggs in the pitcher plants that occur in the bogs around eastern Massachusetts, which is pretty cool. Those don't bite people, though. They don't bite at all, in fact. And so that's another wrinkle that we're interested in. How is it that they get away without a blood meal and able to produce eggs and all? Because what mosquitoes do when they're after blood is females using the proteins in your blood to build eggs. So they're building babies out of blood meals, basically.
Hmm, that's
Yeah.
really interesting.
Yeah, but these guys don't need to for some reason. So I don't know quite what they're using, but they're probably finding lots of protein in the other little animals that live inside those container habitats. You might not find this trait in animals that don't do that. But at any rate, so he's the expert. Sangil Kim and I have collected Tetropes, these milkweed beetles, they're in Mexico. We have a collaborator there, and I collected a lot in Mexico 40 years ago for my own PhD. I froze them at the time. I drove throughout southern Mexico in a little Beetle, a Volkswagen Beetle, which I rented. And I had a container full of liquid nitrogen in the back seat. And I would collect the beetles and put them in that minus 300-degree liquid for the transport back to the United States. And believe it or not, it's some of those specimens that I collected that we're sequencing the genomes of today.
Oh,
40
really?
years later. Yeah, unbelievable, really. I use them for my PhD, but I had enough so that they last. Yeah, it's really quite extraordinary that at those low temperatures, they're still just as if we collected them yesterday. So we collected in the Southwest as well, and Sangil went to many of the same very sites that I collected in the deserts, the Sonoran Desert and elsewhere. And so that's a lot of fun. I really enjoy that part of it. And there I am an expert on collecting insects on plants because I know enough to go to herbaria, to plant collections, to look for recent collection localities for the plants I'm after. And then I go to those locations and usually in the right week, according to the beetle collections that are in museums. And then I'm able to put the two together and find the specimens that I need.
No,
That's been a great formula for 40 years or more. The power of natural history collections, basically.
I know you said that you've always been interested in mosquitoes, but as I know you as the former director of Dr. Class and working with you on the Revista issue, much of your work has been with beetles,
Oh,
including the late David Rockefeller's
right.
extensive collection. What's the difference with working with mosquitoes?
Mosquitoes aren't plant feeders. So for me, the big difference is that if they did feed on plants, they'd be a lot more like beetles. And I could ask the same questions and look on the same plants. Because my work really bounces back and forth between looking at it from the tiny consumer perspective. What about the evolution of host use? And then I turn it around and look at it from the host perspective. Where are my parasites coming from? and what are they doing? And if I was working on mosquitoes, then I'd be interested in everything else that attacks humans. And I am interested in that. So where do the parasites of humans come from? Do they come from so the mosquitoes that attack humans, are their relatives attacking other mammals? Or are they attacking other kinds of animals, other insects or frogs or whatever? And the answers are sensible, actually. They tend to related insects eat related hosts, basically. So there's a narrow pathways to colonizing a new host, and it's usually from something that's similar. Not surprising because evolution tends to be gradual. Which brings me back to stasis, which was your first question, or a middle question at least. My late colleague and dear friend, Stephen Jay Gould, made a career for himself and quite a name from studying stasis. So stasis for him was data, and that's a direct quote. Stasis is data. And stasis, what from his perspective was stasis in the fossil record. He was a paleontologist, and he noticed that for much of evolutionary history, you see the origin of a new life form, and then not a lot happens. So you get a body plan, you got four-legged animals or hard-shelled clams, and then they perk along for 100 million years and they don't change very much. And then all of a sudden, there's a period of rapid change and lots of different kinds of forms might appear. So he thought that there's this pattern of long-term stasis and then occasional punctuated equilibria, which was his term. Punctuated equilibrium is one of the best known phrases in evolutionary biology. And it's this idea that every once in a while, that stasis is punctuated by a period of rapid evolution, which rapidly achieves an equilibrium of, that is, the forms all fit into the existing ecosystems at the time. And then there's another period of stasis. And so he thought that was a real pattern that deserves study. And in fact, he reached way back to R.A. Fisher and said, maybe it's these gene complexes that are involved and that something just has to come along to disturb them to cause new periods of rapid evolution. And nobody took that part of his observations that seriously at the time because we didn't really have any insights into what the genetic mechanism, whether the reality of co-adapted gene complexes, as Fisher called them, but as we call them today, supergenes, accumulations of genes that all encode different aspects of what we call the phenotype, the way an animal lives and breathes or looks or does stuff. Maybe it's a new phenotype for recognizing and then living and reproducing on apple trees. Or maybe it's a new phenotype for attacking humans or living in some place or whatever. So at any rate, I've got very excited by, and I made that connection to Steve's work and what I was interested in, which is long-term evolutionary conservatism, not measured with the fossil record, but studying the fossil record with a rear view mirror, which is using phylogenetic studies to infer the past by looking at the present and variation. Because again, things are conservative. And you know what? It isn't just host use that shows this strong evolutionary pattern of related species being alike. It's all of life because we look like mammals. We look like vertebrates. We look like primates. And it's because we have preserved these traits for a few million, tens of millions, 100 million years that have survived through all of this evolutionary time with so little change that we can still reconstruct that history, not with the fossil record, but by just looking at the comparative anatomy. It's something that Huxley noticed about the primates. It's something that every taxonomist has known for 200 years, and naturalists have known for even longer. So that's slow evolution. And all the money is in fast evolution, the evolution of new stuff. But let's look at slow evolution now that we have the tools to do it. And that's what I'm interested in.
Wow, this slow evolution idea is really interesting, but I'm going to ask you a fast question.
Okay.
Is this slow evolution impeded at all from what you can tell in your studies by climate change?
Let's see. Slow evolution poses a dilemma in the face of climate change because the climate has changed much more slowly than it is today. And so slowly that in the past, animals and plants have been able to adapt to climate change. The evolution of, let me give you the background. So the background is that where we're living today, this what we call a temperate zone, a temperate climate, highly seasonal climate with a period of dry, cold weather is pretty new in the history of the planet. It's like 20 to 30 million years old, which is ancient from our perspective as primates and as humans, but it's really was yesterday as concerns the evolution of vertebrate life or of plant life and the like. And so when that started to appear, and it appeared 20 to 30 million years ago, when the current continents achieve their present place, and that affected the ocean currents, and that affects the climate because it affects the circulation of warm water and other things too, and weather. At any rate, with the current configuration, it posed a dilemma for all these animals and plants because most animal and plant groups evolved in a tropical world. And so only a handful were able to invade and evolve the adaptations for living life in the temperate zone, right? Many plants and animals, for example, migrate south. The animals do. Many plants and animals that don't migrate escape underground. They die off during the winter, or one life stage is built for overwintering underground, and the others don't. So at any rate, now we have that scale of rapid change within hundreds of years, not millions of years. That's a dilemma. In the past, the occasional mutations arose. There was plenty of time for, but yet not every group. We don't have palm trees growing in Massachusetts because palms never evolved the necessary mutations for overwintering, right? We don't have most tropical plants. We don't have most plants. We don't have most animals either. They also can't deal with it. But what we do have did have the enough time to evolve the right mutations. But now we're faced with a claiming changing climate, and it's going to be a new world, of course, in many ways. Of course, but I have to say that the greatest challenges are for humans. It's not for other species. It's for humans because most of our agriculture and most of our cities are at risk because they were built on a model of past climates, not on present climates. What happens when the best grape growing regions are in the Arctic? What happens when the best conditions for growing wheat are similarly in northern Canada? What's going to happen when all of those cities are flooded? That's a real problem. Most 90% of the world population lives close to sea level,
But
um, especially the poorest people. So it's a problem.
with your study of mosquitoes,
Yeah,
as you've said, like the mosquitoes find their places in these recipients of water.
right.
As climate change is producing more drought, does that affect the mosquito population?
Of course it will. They'll move to where water is. On the other hand, it probably will affect the mosquitoes we worry about, which is the ones that are living in cities, less because there are people do have this habit of leaving pots of water around. And it's interesting because there's a lot of science behind mosquito control. I was invited, I think, by Dr. Class, I don't know, 12 years ago or something like that. We did a podcast with somebody from the School of Public Health on climate change and mosquitoes. And there is a lot of research and highly funded research and exciting research on developing vaccines, but also on developing sterilized mosquitoes to control populations. But you know what? The old-fashioned methods of controlling mosquitoes around your home still work best. And those are screens on the windows, fans underneath the tables, no pots of water around. Three things. You don't leave the water around. You keep the fans out from, the fans under the tables because they like to bite naked legs underneath in dark places. And you keep screens on the windows. It's low tech, but I'll give you my little editorial on that too, which I said at the time. And it's that mobilize the high school students as they come out of high school. Their service learning can be educating door to door, their neighborhoods where the people know them. You don't want the Army doing this. You want high school kids to do it. They educate people, go door to door on these cultural changes that can make a big difference in the life of people who are exposed or at risk, basically, to mosquitoes.
Thank
So
you.
there's a public health announcement for you. I
Is
hope Marcia approves.
He's referring to Marcia Castro, who's our wonderful Brazilian expert at the Harvard School of Public Health. Do you have anything else to add about your latest research, Brian?
I think that's pretty good. I have a new postdoc working on amber in the Dominican Republic, so I'm excited about that.
I
Uh,
can't wait to hear about that in
that'll next time.
new podcast. Are you
Yeah, there you go.
listening to Faculty Voices with Brian D. Farrell? Thank you, Brian.
My pleasure, June. Thank you.